Alright, today we're going to be talking about DNA replication, and this is going to go on page 25 in your science log. You may have used 24 yesterday for your notes, and you may not have. We're going to put something on there later. So on page 25, DNA replication, and you need to update your table of contents to reflect that, and today's date is February 7th. So DNA replication, February 7th, on page 25 in your science log. Okay, so replication is to make a copy of something. So when we replicate our DNA, we're making a copy. So you're going to write that down. You don't have to draw the picture. So your DNA is replicated at the beginning of mitosis and at the beginning of meiosis. First, we need to know a little bit about the DNA itself to know how to replicate it. So the shape of the DNA is called a double helix. It looks like a twisted ladder. So that is the word for the shape of DNA, and something's going to have to happen to that twisted ladder before we can replicate it, before we can make a copy. The sides of the ladder is called the backbone. This is a sugar phosphate structure. When we build our DNA model, we're going to use Twizzlers for the backbone because they are made of sugar. And so sugar phosphate is the sides. This is what the rungs or the nitrogen bases will attach to. And the shape double helix was discovered by Watson and Crick. You're going to need to know that later, so make sure you spell those names correctly. They just beat out another team of scientists who were trying to figure out how the molecule was shaped as well. The nitrogen bases are the rungs of the ladder. So that is where you see all the little steps onto the ladder that make up the DNA molecule. Nitrogen bases are going to help form our proteins, and they also make the parts of the DNA. Well, they're not actually going to form our proteins. They're going to tell our proteins how to be made, is more accurate. So the chemicals match up in a certain way, and the A always matches with a T, and the C always matches with a G. So on the strand, there are two actual pieces on that rung, and they combine together chemically like a jigsaw puzzle. So the A kind of piece always fits together with the T. And the A stands for adenine, and the T stands for thymine, and cytosine, and guanine. I'm not going to make you remember those names, but you will learn them in high school. We're just going to remember that the A and the T match up and the C and the G. And one way to remember that is you go to school at Gwinnett County Schools, so AT, at, and then Gwinnett County, GC. Or you can remember them. To make an A, we have a straight line, and to make a T, we use straight lines. But to make a C, we use a curved line, and to make a G, we use a curved line. So you can also, that's another way to remember it. You're going to need to know that. I'll give you a strand of DNA, and I'll say, how would you match this up, or how would you start to replicate it? And you need to know what goes with what. So the first thing that's going to happen is the molecule unzips. So it starts in the middle of those rungs, and it starts breaking apart. And we're going to see a little simulation of this in a minute. And then the nitrogen bases or base pairs are going to start matching up. So if you have an A on one side, it's going to have a T come and match up to it. If you have a T on one side, it's going to have an A come match up to it. So there's thousands and thousands of these that have to get matched up, so it's going to take a little while for that to happen. And then you're going to have two new strands of DNA that are identical to each other. Okay, we're going to take a look at, see if this works for us. So we're going to go to the DNA, and you can do this anytime you want at home. You can do the exact same thing.
Replication is the process by which DNA is copied during the cell cycle. Each strand of the double helix serves as a template from which a new strand is built. Click Start to zoom in and see replication in detail. Enzymes unzip the double helix, exposing the nucleotide bases on each strand and forming a replication bubble. These enzymes unzip the double helix at many places to speed up the replication process. This model, however, shows only one replication bubble. Additional proteins hold each strand apart so replication can occur.
So if you think about how long a DNA molecule is, if we just started from one end and started unzipping it all the way to the other end, it would take a long time. So basically they start opening it at several different places along the DNA strand, and replication is ongoing. So it doesn't have to start from one end and finish at the other end.
Each existing strand of the DNA molecule is a template for a new strand. Nucleotides pair up with the exposed bases on each strand. DNA polymerases bond these nucleotides together to form the new strands. DNA replication is smooth and continuous on one DNA strand. On the other strand, the DNA is made as a series of small fragments that are then joined together. Replication results in two identical double-stranded DNA molecules. DNA replication is semi-conservative. That is, each DNA molecule contains one original strand and one new strand.
Alright, a mutation is a mistake in the genetic code. So mutations can be helpful, they can be harmful, or they can be harmless. So if we look at this DNA strand, what is wrong with it? Take a look and see what's wrong with it. You should see right here the T is matched up to a C instead of an A. Okay, so that is a type of mutation. So if when this was being replicated, if they accidentally put the wrong base in, then that would be called a point mutation. Okay, so mutation is a mistake in the genetic code. Sometimes they're helpful. It actually makes a new phenotype that helps the organism survive. Sometimes it's harmful and it makes something that causes the animal to not be well adapted, or the animal or plant or whatever it is might not even survive because of the mutation. And sometimes mutations are harmless because they're in a part of your DNA that never gets read. So if it's not in an area where you're making a protein from that part of the DNA, it could just not do anything at all. Okay, you're going to be watching this video on types of mutations. You're going to follow along with the guided notes. Oops, sorry. So there's going to be a little half sheet of paper you're going to get, and you're going to watch Mr. Anderson talk about mutations, and he's going to show you all the different types of mutations. And as you watch him talk about them, you're going to write down the things that he's telling you, following along on your guided notes, which are going to go on page 26 or 25. Let me look and see my science log. 26. Those notes are going to go on 26, and you're going to have a piece of paper to write along with. It has a front and a back, so make sure when you get to the end of the video, you're probably going to have to flip the paper over. Okay?